the organization of the meeting. Thank you for putting this effort together. And thank you for giving me the chance to talk about the EM platform at the IGBMC in Strasburg. So the IGBMC, is this working? Okay. The IGBMC is the Institute for Genetic and Molecular Biology and Cellular Biology in Strasburg, and hosts the Cryo-EM platform in which I am involved. So, to put things in context, I want to introduce the Center of Integrative Biology, CBI, which is part of the Institute, the IGBMC, but is an entity for itself because it was created with large support of the French government and local government to create a center in which many cutting-edge technologies can be offered to the French community. And in the Center of Integrative Biology, we have a platform for molecular sample production and characterization, X-ray crystallization and X-ray crystallography. We have super resolution microscopy and also, of course, EM. And this effort to put all this platform in this building is what we call the Integrative Structural Biology platform. It will allow us to give access, also participate in infrastructures like Instruct or the French infrastructure for integrative structural biology called FRISBI. So I wanted to talk about this is because you have here, I wanted to talk about the users of our platform. We receive users from the Institute in all these dependencies. It's more of 10 internal groups that are regular users from the platform, and with different intensities. There are a couple of groups that have tradition in Cryo-EM, and others are directed by crystallographers who are starting to move to EM or take advantage of the technology. But not only this large number of groups are working with us, we receive users from Europe through Instruct-ERIC and from French groups through this platform that I mentioned before, FRISBI. So, as you see, well, we have to deal with this high demand of projects and groups. And the offers that we offer are listed here, everything related with Cryo-electron microscopy. We do sample preparation. The pre-screening in negative stain is used in many cases in initial stage of the project. Then we do vitrification of the samples using microwave and pre-screening in cryo conditions. This is to assure that the samples are sufficient, good enough for the high-end microscopes and final data collection. We offer data collection in two different techniques, and as I know that not all of you are in the field, I just wanted to mention very quickly that in single particle analysis we take images of relative homogeneous samples embedded in ice, like the ribosome here in this illustration, and by combining thousands of images in different orientation, you can achieve a very high resolution structure of your macromolecule. The other technique is cryo-electron tomography. I will talk later about it. These two are the ones that we favor. And after we have collected data, the analysis of the data is carried out by the groups itself. But we offer pre-processing of the data, very easy steps at the beginning, which are common for all the projects. The instrumentation, we have basically 3 instruments, one Titan, one Polara, and one Technical F20. The first one is the high-end microscope, and is the only one that we have, only in quotation marks. We are happy to have such an instrument, but it's only one in comparison to other facilities, larger facilities that have several of them. However, it's a very good equipment. It's equipped with energy filters, CS corrector, Volta-Phase Plate. We recently, or last year, we go through a process of upgrade the system with K2 Submit camera and Falcon 3. I just arrived when we were doing the acceptance test of the Falcon 3, and the microscope has also capabilities for dual-axis tilt tomography. In the Polara and the Tegna, we use this microscope more for the screening of the samples in cryo conditions. And recently we take the Tegna to Falcon 2, so probably we are going to use the Polara also for some specific projects. This is some of the achievement in this period 2013 or 14 in terms of structures. We published several structures. Probably the highest resolution was of the human AES ribosome at 3.6 angstrom resolution, more or less. And these are complexes that have to do with related with gene expression. They are nucleic acid binding complexes like nuclear receptors, transcription factor, GRAs, or the specific project was done in Ribosome using tomography. But after that, there was this period of upgrades of systems, and more recently published this at the end of last year. We had the group of Dr. Clajos published this very nice structure of 80 ribosomes at a very high resolution, up to 2.5 angstrom resolution, to identify and map all chemical modifications of the 80 ribosomes in the nucleic acid. So, for example, you have methylations of some guanidines that are visible only at this high resolution, or some pseudo-pyrimidines, which can form hydrogen bonds with neighboring ribosome, stabilizing RNA helices. So this work is important because it has significance in drug development and understanding the modification of the function of the modification in the human ribosome. Something still more recent. This work was done with still the Falcon 2, and something even more recent published last week is from the group of Albert Baxel-Balima. He studied the one complex of the RNA polymerase in connection with a protein factor called Nusa, which stabilizes transcriptional ribosome. So they solve the structure of two complexes, E. coli RNA polymerase, one to Nusa, without a hairpin, one of the structures involved in the pausing of the RNA polymerase during elongation, and achieving resolution of 3.6 4 angstrom resolution using the K2. So this is one of the first structures that are published now with a new camera. The work has been well received, and they got the covers of the last issue of Molecular Cell. It's very funny, so it represents the RNA polymerase here as cowboy, one running during the elongation process and one resting, with the hot symbolizing this Nusa factor which stabilizes the polymerase at AP. So good. Sorry. This is what had happened until now and before my arrival, and in a few weeks we expect a couple of other structures that have been solved in the time when I have been in the institute. one of these points in the points that I want to make in this process of optimizing the resources that we have is the upgrade of the cameras. But the next point is the use of Volta-Phase Plate. So when I was postdoc student in the Department of Dr. Baumeister, one of the recent developments was the Volta-Phase Plate driven by Rado. He helped Dr. Cajos in Strasburg to set the technique, and this is an example of ADX ribosomes. The Volta-Phase Plate is a device that helps increase the contrast of the images when you are very close to focus. So this is a comparison with the normal conventional technique of the focusing as -3.5 micrometer focus, and with the use of phase plate. You see immediately that the contrast is much more higher. And maybe less clear, but also very significant, is the use in cryo-electron tomography. These are examples of bacteriophages, and here you can see that there is more structure, there is more dense than with the conventional techniques at the focus of -6 micrometers. So the phase plate really is something that I think will have a major impact in the field. And Dr. Clajos made this work published also this year, showing that the Volta-Phase Plate can be useful not only in the applications of when you are trying to solve very small complexes, less than 100 kilodaltons, but also in general, because here this is an example of ADX ribosomes imaged with and without phase plate. And what he tried to emphasize is that the use of phase plate facilitates the processing of the data. This is an example of 2D classification and averaging of the projections. And you see that this process of classification and alignment of 2D projections is iterative. And with the use of phase plates, you get much more, the images converge much more faster than when you don't use phase plate. Also, the final assignment of angles for each particle is much more fine. And as a consequence, you can achieve reconstruction with similar resolution using more or less a half of the number of particles that you will use for conventional methods. So actually that is quite interesting for us, because when you reduce the number of data, you are going to be more efficient. So in terms of software, we use the most common software used at the moment. EPU and Tomo4 are the commercial software provided by FBI and Thermo Fisher. EPU for single particle, Tomo4 for tomography. And the other alternative is IGBM, which originally was created to do tomography, but it's also worked very fine for data acquisition for single particle. And actually, our preferred software at the moment is IGBM. Basically because it's faster. So when you compare to the version that we had, we installed in October of EPU, CLEM is 20% faster, more or less, than EPU. And the new version of EPU was released 2 weeks ago. We are going to get it and test it soon. I think they get a significant improvement of speed also. So one of the things that I like from EM is a feature that is also present in EPU, but here we have it also in CLEM about multi-shot functionality. So we can take many images per hole. The samples in EM are in holes in a plastic or irregular holes. Okay. So in this case, I don't want to go too technically, but this is an important feature. You can take many images with only one cycle of the focus, and this is one of the features that allows us to take about 95 images per hour in super resolution mode when we save images in TIFF. And as a consequence, we have something like 3.5, 4,000 images in 40 hours, which is a session of 2 days. No? And this is the rhythm that we have implemented in the last months, okay? Sessions of 2 days for each project. Well, I don't know if all of you are concerned that that in the recent times, when you take an image at the microscope, you don't take just one picture. You collect what we call movies, a collection, a stack of images, very short exposure times, and that allows to make a preprocessing in which we align frames to correct for a small movement, pin-induced movement of the sample. And with this trick, you create a picture that is much more sharp and it has potential to give higher resolution. And this preprocessing is what we include in our preprocessing pipeline on the fly. The way in which it's implemented at the moment is that images that are collected at the title are saved by CRDM in a stack of 20, 40 frames in super resolution mode in TIFF format. So this is important to save space. They are compressed. One image or one stack is 250 megabytes. If you were using not compressed files, it would be several gigas. Then these images are transferred to another PC in which the users can introduce a very simple interface parameters for the motion correction. And actually, it's a dedicated preprocessing PC with 4 GPUs, which is responsible to run the jobs of motion correction. Here, for example, we start a micrograph can be processed in 37 seconds. And as the camera has the potential to process 2 micrographs per minute, we can cope with the high data acquisition rate of the microscope. The motion corrected images are also analyzed by the program of CTF estimation. And these results are visualized in such a way that can also help to modify the parameters of acquisition on the fly. No? So this is part of the Instruct-ULTRA project, and I know that there are other alternatives very similar to this project. And one of the good things of this meeting is to interact with people from, for example, Scipion, which has similar capabilities and other tools. So this is a way in which it looks over the repository pipeline. It's a novel framework in which they try to create something in which user can introduce very easily through a very easy, user-friendly GUI all these parameters and create on the fly processing workflow, which can be portable, parallelized, can allow remote access, etc. This is the work of two good programmers, Jonathan Michelin and Luca van der Velden. I won't talk too much about tomography because most of the work that we have done at the moment is single particle. I just recently started to make tomography. I would say that is more close to my heart because I was doing tomography in the department of Volta-Phase for many years. And these are examples of the work that I have been done. So in tomography, you don't require only single projection. You kill the specimen inside the microscope and take kill series of images. And with these different views of only one object, you can look through a three-dimensional volume. And this allows to study pleomorphic structures, a structure that cannot be averaged, but also if molecules are repetitive, are found in those tomograms, then you can do some tomogram averaging and produce also structures. And these are some of the examples that we published in the past related with supramolecular organization of ribosomes. Dr. Gah's group did a similar thing in ADS ribosomes, the human ribosomes. So we want to go in this direction because with new technologies like the feed milling technology that we are also implemented, I think Julie will talk about that in the next talk. I think we are going in this direction of recognizing molecules inside the cell. Sorry. For conclusions, then the improvement of data quality, analysis of smaller complexes, high resolution, and high throughput are achieved by upgrading hardware, direct electron detectors and Volta-Phase Plate, developments in data acquisition in the software, so better data processing of Volta-Phase Plate images, and this on-the-fly processing. There is a continuous optimization of image settings, playing around on how many cycles you use for each activity. And something that I haven't talked about it, but it's very, very important, is the commitment of the platform power to cope with a demanding booking system of today's sessions. So we have a limited number of people that are able to handle the microscopes at the moment. Well, when I started, we're only 3 persons, including me, and now we are training more people. 2 new students are already trained, and during this year there will be a group. My expectation is that every group, internal group at the institute, have at least 1 member able to work alone independently at the time. For the outlook, I would like to say that we would like to promote inter-platform networking, and it's nice these kind of events to get in contact with the responsible for the facilities. We would like to work in the open remote access through Instruct-ULTRA. And we are working on the creation of a French National Center for Biomedical Cryo-EM. It's a project in which many institutions has cooperated, and we has a very positive perspective to come to a good end at the end of this year. And it's about to acquire at least 1 title for our additional title for our center, and to buy another microscope for sample optimization in other places in France, these red points, equip existing microscope with direct electron detectors, and expand the capabilities in cell imaging and computing in several centers. So I hope that with that, Cryo-EM can become stronger in France. About the people, well, I want to thank all my colleagues here. The platform itself is comes with the administrative coordinator, Michael Pottersman, Bruno Caus and Ernesto Poyani has scientific coordination roles. Gabor Babay and Clarotilia help me with the Titan acquisition. Jean-Francois Corinne Crucifix are more involved in sample preparation and screening. They have a very tough job. Igor and Daniel are working with cryo-feed milling. So thank you for your attention.
That's great. So, questions from people?